Hi everyone, I'm Dr. Ellie Huang. I'm a scientist and curriculum team member at Mini PCR Bio, and I'm excited today to share with the IBE community my work on applying cell-free technology to education. So I'm probably preaching to the choir here, and that we can all agree that biology education is really important, and that scientific illiteracy is a growing problem in our country with the general public, people who don't have a science background, not understanding the basic science behind things like GMOs, evolution, or vaccines. And speaking of vaccines, the pandemic over the last 2 years has really highlighted just how little the general public remembers about what they learned in school about things like DNA, RNA, and protein. And so they don't know what the mRNA vaccine does and how it works. And this is actually leading to a lot of problems in terms of public health. So when we look at the activities that we can actually do in the classroom level at the molecular biology level, there are actually quite a few activities that are being done now in classrooms. Things like transforming bacteria with a plasmid that encodes GFP, so you can see your cells light up to understand DNA, RNA, protein, how bacterial transformation works, to gel electrophoresis, so you can understand how you can separate DNA by size and how that's a really useful application. All these hands-on tools are really great for students to really get and understand all these basic molecular biology ideas. However, there are a lot of challenges in implementing these hands-on molecular biology labs in classrooms. The biggest is that, it can sometimes be hard to see and understand what is happening. A lot of molecular biology, as we know, is just pipetting a lot of clear liquids into other clear liquids into really small tubes. And for a student doing this for the first time, they may not understand properly what's actually going on. These labs can also, require really expensive equipment. If you want to do bacterial transformation, for example, you need -80s to be able to keep your cells stable. You need shakers, you need incubators, you need, PCR, gel electrophoresis equipment, all of that. And that can be out of budget for a lot of schools. And finally, these labs can be quite complicated to run and does require a lot of teacher prep time or expertise that teachers simply just don't have right now. So when I started my PhD at MIT, I joined the Jim Collins lab, and one of the things that the lab was working on was this idea of cell-free protein synthesis. So I'll just run through this process really briefly before diving into how I applied it to education. So what cell-free protein synthesis is this idea that you can actually extract the necessary components out of live cells and use them in a cell-free system to be able to still express proteins. So basically you would grow a bunch of cells like E. coli. These would be the normal cells that you would use for live bacterial transformation to make proteins, for example. Grow them up, harvest them, and then you would actually lyse these cells, kill them, and basically scoop out all the molecular machinery inside, things like the polymerases, the ribosomes, and as long as you add the proper cofactors and substrates, an energy sort, if you add a DNA template into this reaction in this tube, it's going to make whatever protein the DNA encodes for. The advantages of this is that it's a abiotic reaction, meaning that it's not alive, so you don't have to worry about keeping any cells alive. You don't need shakers, incubators, and -80 freezers. The other advantage is that it's an open reaction. Instead of having to, jam a plasmid through the bacteria through transformation, it's directly open, and you can actually manipulate this reaction a lot easier, which makes it very useful in terms of education. And in the Collins lab, they were actually freeze drying these reactions, which is really cool. The concept of freeze drying is really simple. Imagine you had an ice cream cone. If you just let it sit out at room temperature, it's going to melt. But you could dip it in liquid nitrogen and use a special machine called a lyophilizer to suck out all the water, resulting in just a solid chunk of freeze-dried ice cream left over. Now it's stable and it's not going to go bad. And we can do the same thing to these cell-free reactions. So you can see these yellow pellets at the bottom here. Those are the cell-free reactions that have been freeze-dried. Now they're shelf-stable, which makes them a lot easier to ship and store. So in the Collins lab, there were a lot of work going on with freeze-dry cell-free applications being applied to things like diagnostics, for example, freeze-drying the reactions onto little paper discs with a biosensor that can detect Zika virus. And so if you put a patient sample on here, if it turns purple, you know the patient sample has Zika. If it's yellow, it's negative. The advantage of the diagnostic like this, that it's rapid, it's cheap, and you can easily transport it to the point of care where you need it. People in our lab also worked on freeze drying these reactions onto fabrics with the thought of, we could create a doctor uniform that could detect superbugs in the hospital, or onto soldiers' uniforms so they can detect biochemical warfare while on the battlefield. And so when I joined the lab, I thought this technology was really, really cool. And what I realized was, besides the fact that they were cheap, easy to use, and quick to use, it was actually really distilling molecular biology down to its fundamental ideas. In fact, back in the 50s or 60s, when they were working out how the central dogma worked, how does transcription, translation, specifically translation, they were trying to work out. These two German scientists actually used cell-free extracts to figure out the genetic code, and they actually won the Nobel Prize for this work in 1961. And so it's a really great demonstration that these cell-free systems are distilling down these molecular biology ideas to their fundamentals, which makes it easy to work with in a research setting, but also really makes it a useful educational tool. Add on to the fact that it's cheap and easy, makes it something that I thought would be really great for the classroom. So in the course of my PhD, my grad school thesis was this idea of developing biobits, which was these freeze-dried cell-free reactions specifically for educational uses, preparing these activities for students to be able to do easily a wide variety of molecular biology activities in the classroom. My advisor, Jim Collins, my lab mate, Peter Nguyen at the Wyss Institute, teamed up with Mike Jewett's lab at Northwestern with Jessica Stark as the lead grad student there, and we formed Team BioBits. And together, we prototyped a really wide range of different activities with this idea in mind of creating some sort of kit where you had these freeze-dried cell-free reactions that you could add, plasmid DNA into that encoded a bunch of different proteins, and that these proteins would have outputs that were educationally engaging to students. Things that they could see, like fluorescent proteins or colorimetric proteins, enzymes that could generate compounds that they could smell, enzymes that could generate hydrogels that they could poke, and even getting into things like programmable synthetic biosensors to get into synthetic biology topics as well. So with fluorescent proteins as a visual output, this was a pretty low-hanging fruit, right? We were easily able to express a range of different fluorescent proteins in the cell-free system, and this can be used by teachers to demonstrate things like protein expression, gene expression, the central dogma to students. We can also give students a little bit of opportunity to play around with the reactions and get excited about it. Here are, I think, plates with freeze-dried reactions at the bottom of each one, and students were actually able to come up with their own design, decide, okay, if I want to make this design, what plasmid do I need to add to which well to get a rainbow or a periodic table? And these are actually pictures that actual students created with the cell-free reactions. We also went on the other end of the spectrum and went for advanced topics to teach synthetic biology. A common lab activity that's done in classrooms right now is taking fruit like a strawberry, a banana, or a kiwi, squishing it up with some soap, and then precipitating the DNA out with isopropanol, and you get this goopy DNA at the end. And it's a cool demonstration, but the lab stops there. So we decided to use BioBits to extend this activity even further. So students could actually take the DNA that they extracted from the fruit, amplify it up, and stick it onto stick it with a BioBits reaction that had a sensor that was specific for certain types of fruit. And so you can see here that if they stick the banana DNA onto the banana sensor, it glows green, but not when you put kiwi or strawberry. And same thing with the kiwi sensor. If you put kiwi DNA, it glows green, but not when you put the banana or strawberry DNA. So a great example to students about how synthetic biology could be leveraged to create things like diagnostics. We also came up with a bunch of activities to teach quote unquote hot biology topics that teachers really love bringing up in the classroom, things like antibiotic resistance, the mechanisms behind it, as well as CRISPR-Cas. So if you want to see all these details of these different prototypes, we did publish them open source. There are 2 papers in Science Advances, as well as a paper in ACS SynBio. If you just put in BioBits into PubMed, these 3 papers will come up, and you can read through the details about all these different prototypes. But when I graduated with my PhD, I didn't want to leave BioBits behind. I actually wanted to turn these prototypes into an actual product that teachers and students could use in the classroom. And so the next step with what I did is I actually interviewed a wide variety of high school biology teachers to actually find out like what are things that I'm missing that I need to know in order to develop these activities further. And I learned a lot of things, and I'll share 3 of them, the 3 big ideas that I learned. One is that students are not the best at following directions, especially when they're operating things that are unfamiliar to them, like micropipettes. And so the lab protocols need to be simple and robust, trying to minimize pipetting volumes, try to make pipetting volumes as big as possible, making directions very, very clear, with the goal of being able to have an activity that is simple enough for the students to do that they don't lose sight of the science behind the activity that they're doing. Teachers also have very little time and money to teach all the required standards. Unfortunately, teachers are given a laundry list of topics they have to cover within the school year, and so they really prefer hands-on labs that will teach these fundamental concepts and check off these standards. So, which means that unfortunately, labs that teach cool things like synthetic biology or diagnostics may not be as popular as a lab that would teach something like the central dogma. So that was something to keep in mind. And finally, that the accompanying curriculum to pair with the lab is just maybe even more important than the lab activity itself. Even though we're using fluorescent outputs to be able to, highlight what's going on, students still need a little bit of help understanding what's going on at the molecular level. And so the curriculum, the lab guide, the study questions, the background that come with the lab are very important. You need to be able to communicate to the students what is happening in the reaction tubes that they're doing. So I decided for the first lab we were actually going to try to take to product would be the central dogma. So this is a lab that I developed where the DNA that they add to the freeze-dried cell-free reaction is shown here in RNA form. Basically, there is a part of the DNA that codes for an aptamer. This aptamer, when it's in RNA form, folds up and binds to a fluorophore that emits a green fluorescence. So in this case, when students see green, that will indicate that they have RNA and that transcription is successful. The DNA that they add also encodes RFP, a red fluorescent protein, when they see red, they know they have that red fluorescent protein and that it indicates translation. And so they're able to very quickly see the green fluorescence indicating transcription after just 10 to 15 minutes. And if they come back the next day, as the red fluorescent protein needs time to mature, they're actually able to see the red the next day. And as part of this lab, we also threw in these other tubes. The reason why tubes 3 and 4 don't have red the next day is because in these tubes, we add things like antibiotics to inhibit translation. So we show students that transcription and translation are 2 different processes, and you can actually inhibit translation without inhibiting transcription. And so that's just another piece of curriculum that we threw in there in the lab. So here's a video that kind of demonstrates. This is obviously very sped up, so you can see it's green, and then it fades out eventually to the red fluorescent protein. So visual representation of the central dogma, DNA to RNA to protein, the flow of genetic information, to transcription and translation with these color indicators so they know what part of the process they're dealing with. So I brought all this work to Mini PCR Bio, which is a small biotech education company based out of Boston. They make a lot of biotechnology equipment that is accessible, as well as really innovative curriculum for teachers to be able to use that equipment in their classrooms. And it was really a great match for me to bring this work to them. They had the same mission statement as me, making science accessible for everyone everywhere. They're already well established in the biology education technology space. I didn't necessarily want to spin my wheels and try to figure that out for BioBits myself. My main goal was just to get this in the hands of teachers and students, and so teaming up with someone already well established in that space will allow me to get to that goal quicker. They were also experts in curriculum development, which I am not. And as I mentioned before, curriculum development is extremely important with these labs. BioBits fit in their portfolio very well as they were looking to move into protein biology. And they actually had already developed the hardware, a blue light illuminator that was, cheap and easy to use for students that they could visualize their finished BioBits reactions in. So brought all the BioBits work that I did in my grad school to them. And with them, I was actually able to take the central dogma lab from the prototype to a real activity, from overseeing product design and production and actually producing these kits, to seeing students use them in classrooms and sharing their pictures of the results on Twitter, to even during the pandemic, teachers were actually adapting the central dogma lab for at-home learning because it was shelf-stable and didn't require a lot of equipment, just that viewer and a pipette. They were actually able to send these kits to students at home for them to do the reactions together all over Zoom, so they could still continue doing hands-on learning, which was really cool to see. And we didn't stop there. BioBits is being used not just in the US, but all around the world as well. Here's a picture of a teacher development workshop in Japan where they're teaching BioBits. BioBits has been translated into German to be used in schools there. And I believe this is a video in Turkish of someone demonstrating BioBits as well. And in just a few months, actually, we are going to be sending BioBits up into the International Space Station for scientists to be able to do cell-free reactions up there. This is part of our Genes in Space contest, which is a free contest we run every year. And the winning experiment from this year's contest was a student proposal that is going to be using BioBits as a sensor aboard the ISS. I'm very excited to see my project get launched up to the International Space Station and see what the results of that student proposal will be. I'd like to thank Team BioBits, Jim Collins, Mike Jewett, Jessica Stark, and Peter Nguyen, as well as my Mini PCR Bio team. And if you have any questions at all, please email me at ali@minipcr.com. I love talking to researchers about how they can translate their work with an educational angle, and love talking about ideas for outreach as well. Thank you.